Okay.
Okay, can you see my screen? Okay.
Yeah.
Okay, so just a quick overview of what we're going to be doing. So this is session 6, which is gene expression. So I'm going to be starting the session with prokaryotic gene expression. So I'll be covering operons. So we'll go through a simulation of the lac operon. We'll talk a little bit about DNA looping and ribo switches, and then we'll have a quick break, and then I'll hand it over to Shruti, who will be talking about eukaryotic gene expression and some techniques. Gene expression is regulated through genetic switches, which is the concept that genes can be switched on or off in response to signals. Example that we'll be going into detail with is E. coli adapting to changes in their growth media. The logic here is that only the genes that are necessary for the present environment would be expressed. So the cell isn't wasting resources expressing genes that expressing genes that are not necessary at the moment. I'm going to focus on prokaryotes now because they're generally simpler than eukaryotes. In prokaryotes, regulation of gene expression primarily occurs at the level of transcription. As you probably remember from session 1, transcription is the flow of information from DNA to messenger RNA. So some key features in prokaryotes are that genes are organized in clusters that are called operons. And transcription of one of these gene clusters is controlled by a single promoter onto a single mRNA molecule. So an example here is a tryptophan biosynthesis operon, or the TRIP operon. So you can see there are 5 genes that are involved in tryptophan biosynthesis, and they are under the control of one promoter, and they're also associated with one operator region. So operons can be controlled both by positive and by negative regulation, both a positive and negative regulation is carried out by binding to ligands. So an example of negative regulation, an addition of the ligand can switch the gene on by removing a repressor protein, or a removal of the ligand can switch the gene on by removing the repressor protein. Similarly, in terms of positive regulation, an addition of a ligand can switch the gene off by removing an activator protein, or removal of a ligand can switch the gene off by removing the activator protein to turn the gene on. So a good example of negative regulation of an operon is the TRIP operon. A bacterial cell can obtain tryptophan from the environment, or it can, in the absence of environmental tryptophan, it can synthesize it itself through activation of the TRIP operon. So in the presence of high tryptophan levels, a repressor is present, and it binds to the operator region of the TRIP operon. So this prevents RNA polymerase from transcribing the coding region of the operon, which is located beyond the operator region. So this operator region is in between the promoter and the coding region, and a repressor bound to a co-repressor, in this case tryptophan, then binds to the operator, and this physically prevents RNA polymerase from continuing transcription of the coding region. In the absence of tryptophan, this repressor dissociates from the operator region, and RNA polymerase is able to synthesize RNA at this operon. An example of an operon that's controlled both by positive and negative regulation is the lac operon. So the lac operon has three structural genes that it codes for. One is beta galactosidase, which converts lactose to, it can either convert lactose to allolactose, which is metabolic intermediate, or it can cleave lactose to galactose and glucose. The reason why the cell would want to do this is because the preferred energy source for bacteria is glucose. But in the absence of glucose, the cell needs to activate all operons to allow for utilization of alternative sources of energy, such as lactose and arabinose. So the lac operon converts lactose to galactose and glucose so that the cell can use this for energy. So like I said, one of the genes in the lac operon is codes for beta galactosidase, which carries out this enzymatic reaction. Another enzyme that's coded for in the lac operon is lactose permease, which is involved in the transport of extracellular lactose into the cell. And the last gene is transacetylase. This one, the function is still currently unknown. Other features of the lac operon are the lac repressor, which is upstream of the lac of the promoter region. So this gene comes with a separate promoter, and this codes for a repressor protein, which is represented here. So this is a repressor protein that binds to the operator region, and this prevents RNA polymerase from transcribing the three structural genes in the lac operon.
Can I just ask a question here? Mm-hmm. So is the lac operon for each of these different genes, for beta galactosidase, for allolactase, or does each gene have a different operon, have a different lac operon, or is there one lac operon and then within it are all of these structural genes?
It's one lac operon, and all three of them are part of it. All three of these genes are.
That's helpful because you usually need all of the genes together in a bucket to do a thing, right? So if you can regulate them all together, then you have production of all three at once. This is very different from the way that our genes work, but bacteria have a much more efficient system.
Well, since you're going to end up with one mRNA, and then the ribosome is going to come along and translate that, it's going to produce a continuous polypeptide chain. So are there enzymes that cleave it, or is it self-cleaving? How does it separate into the?
There's signals in the mRNA that direct the ribosome to stop translating at when one protein is completed.
So you have stop codons after each one, and then another start codon, or it works differently?
Yeah, I think that is how it works.
Yeah,
if I'm remembering it correctly.
And I see Shrudi's asking about the IRES, and I can't remember what that stands for. Internal ribosomal entry site.
Yeah, that's what I remember. I don't work with prokaryotes, so I don't. My memory of this is super vague, but I do know that we've used it in my previous lab for like a reporter plasmid. So I don't know if that's a feature of the lac operon or if that's like a feature from another bacterial structure that we were using. But I think it is like a feature of operons, right? Like that you have your continuous thing and the ribosome knows where exactly in that chain to go to start translating.
Yeah.
Maybe.
So it's basically multiple ribosome binding sites. So the mRNA is out floating around there. The ribosomes can associate with any of the the binding sites, and we'll just translate that one gene. That one gene for that one protein. The reason I ask is because we're in Open Plant, we're engineering a reporter gene that is a biosynthetic pathway that's going to produce the same red that's in beets. So you know, you can see it easily, and that's actually a self-cleaving peptide. So, which I don't exactly understand how it works. So it breaks apart by itself. So that the ribosome just chews through the whole thing and produces the polypeptide chain, and then it self cleaves.
Okay, so I'm going to go into the repression mechanism first, and then the activating mechanism. Yeah, so you can see here that that the LAC-I region codes for the LAC repressor, which binds to the operator region, and this binding occurs only when lactose is absent. The reason why this doesn't happen when lactose is present is because lactose or the intermediate allolactose will bind to the repressor protein. This causes a conformational change that results in the repressor being released from the operator's region. And this will allow for transcription by RNA polymerase. Conversely, there's also positive regulation at the LAC operon, and this occurs during conditions of low glucose. So when glucose levels are low, the cell accumulates cyclic AMP, which is a binding partner of a protein called CAP. So the CAP protein, when it's bound to cyclic AMP, binds to what's referred to as a CAP site, which is upstream of the promoter, and this promotes the interaction between RNA polymerase and the promoter, and this enhances transcription of the LAC operon. In conditions of high glucose, the enzyme that converts ATP to cyclic AMP is inhibited. So the CAP protein doesn't have its cyclic AMP binding partner. So transcription can still occur, but it doesn't occur as efficiently as it would in low glucose conditions. So in summary, the conditions in which the LAC operon is activated is it's only when there's low glucose and high lactose. The reason for that is in high glucose conditions, there's no binding between the CAP protein and the LAC operon, and in low lactose conditions, the LAC repressor is bound to the operator sequence. So I'm going to go through a simulation of this now so you can visualize it better. Okay, so this is a visualization of the LAC-Z gene being transcribed. So LAC-Z is beta galactosidase, which is the enzyme that breaks lactose down into glucose and galactose. So you can see here, LAC-I is being transcribed. That codes for the repressor protein, which binds to the operator of the LAC-Z gene. And if you add lactose to the system, it binds to the repressor and removes it from the LAC-Z operator. And this enhances the ability of RNA polymerase, which is represented by this, to transcribe the LAC-Z gene, which is represented by this structure, and you can see it breaking down lactose to glucose and galactose. And this simulation shows lactose transport into the cell and how that would affect transcription at the lac operon. So here we're going to look at both LAC-Z and LAC-Y. LAC-Y is the lactose permease enzyme. So that is the enzyme that allows lactose into the cell from an extracellular source. So you can see right now the repressor is preventing RNA polymerase from transcribing the operon. And see, this is a new mRNA strand that is being translated to LAC-Z and LAC-I. LAC-I is allowing lactose into the cell, and that is causing the repressor to be removed from the operator. Right, so the link's in the slide, so if you want to check that out on your own, you can access it there. So I want to talk a little bit about the structure of the LAC repressor. So this is the crystal structure. The LAC repressor is a homotetramer, which means that it consists of 4 subunits that are identical, and each subunit has 4 functional domains. So those domains are an N-terminal DNA binding domain, which has a characteristic helix-turn-helix motif. It has a flexible hinge domain, a core domain, and the core domain is what undergoes conformational change when it binds the inducer, which is allolactose in this situation. And there's a C-terminal alpha helix, and all 4 of these for all 4 of the subunits, the C-terminal alpha helix will associate with each other, and that is how the quaternary structure of the LAC repressor is generated. Through the use of DNA's footprinting, it was determined what sequence in the operator region is bound by the repressor, and it was found that each pair of subunits within the repressor binds to one operator sequence, which means that the LAC repressor homotetramer complex interacts with the operon at 2 separate sites. This creates something called a DNA loop. So DNA looping is when a protein, specifically a transcription factor, simultaneously binds onto a stretch of DNA at 2 sites, and then the intervening DNA is looped out. You're going to see a similar mechanism in eukaryotes as well, but as with most things, it's more complex in a eukaryotic system. So DNA looping can be used to repress transcription or activate transcription. So in panel A here, they're showing transcriptional repression. So here RNA polymerase activity is blocked. This can occur by competition for binding sites between RNA polymerase and a transcription factor, or it can occur by trapping the RNA polymerase in the DNA loop, or it can also result in prevention of recruitment of an activator. And in B, they're showing activation of transcription by DNA looping. So this is also referred to as an enhancer, and this may facilitate binding of RNA polymerase to the stretch of DNA. And the last mechanism of regulation of gene expression in prokaryotes that I want to talk about is riboswitches. So riboswitches are sequences within mRNA that can bind metabolites or metal ions as ligands, and this causes them to adopt a alternate secondary structure. So this can occur after binding to things such as magnesium ions and enzymatic cofactors or nucleic acid precursors. So riboswitches, they're usually present at the 5' untranslated region, which is upstream of where the start codon is, and they typically consist of 2 domains. One is the aptamer domain. This is the region that actually binds the ligand, and the other domain is called the expression platform. So this acts directly on gene expression through changes in a secondary structure. And the switching sequence is shared by the 2 domains, and this is where the secondary structure is can potentially be altered. So the 2 types of secondary structures that are shown here are an anti-terminator loop, which prevents the formation of a terminator stem loop, or alternatively a terminator stem loop, which signals termination of transcription. So there are multiple factors that can affect riboswitch function. This can include the rate of riboswitch transcription, including pauses in transcription. The rate of mRNA structural rearrangements can also affect riboswitch function, and the rate of ligand binding can also affect riboswitch function. So all of these are examples of kinetic control. After the transcription is initiated, because the aptamer region is in the 5' untranslated region, it's typically the first thing to be transcribed in an mRNA sequence. After this initial transcription, there is sometimes a pause after transcription initiation, which is represented by the star here. At this point, if the aptamer binds to a ligand, this can result in the formation of a terminator stem loop. So that is represented by this hairpin like structure, and this signals the end of transcription. Alternatively, if there isn't a pause after, if there isn't any binding to the ligand after the initial pause, the transcription can proceed as normal. Typically, there's another pause in the middle of transcription. At this point, if binding to the ligand doesn't occur, you have the formation of an anti-terminator loop, and this prevents the formation of a terminator stem loop and ensures that transcription will be completed. There are several kinetic factors that are involved here. So Kf is refers to the rate of the secondary structure alterations in the aptamer. Ktx refers to the rate of transcription, and K observed and K un represents the rate of aptamer ligand binding. So this is something that can be affected by the ligand concentration in the cell. So in order for riboswitch to terminate transcription, the concentration of the ligand has to be higher than the rate of transcription, because if transcription proceeds beyond this initial pause without binding, there won't be any formation of the terminator stem loop. So any questions about riboswitches?
Yeah, I'm not getting how the terminator loop causes transcription to end. Is it formed after the ribosome has passed by that region?
No, it should be formed prior to that, and also this multiple uracil sequence also signals the end of transcription.
Right, but that was there already, right? because it's part of it's this loop that gets formed that causes the termination.
Yeah.
And what is the loop that's on the bottom here, the loop that points down that says AT? Why is that an anti-terminator?
I think the logic is that this alters the mRNA structure so that the RNA polymerase will reach the polyuracil sequence before before transcription is completed, whereas here that structure isn't formed.
I was just looking for the presentation you sent. I didn't see the link to the animation.
Okay, I'll include it in the chat box.
Okay, great, thanks.
One question I have about these various ways of controlling gene expression, are some more frequent than others? Are ribose switches, do they happen 10% of the time? And is it operons the other 50%? And what's that distribution like?
I don't think they're exclusive of each other. So I'm not sure exactly how frequent each is, but yeah, I'll have to look into that.
Yeah, my best guess is it probably varies by species too, because it's all sequence dependent, right? So so probably it's hard to like make a like clear assessment of that, because each critter is going to have its different evolutionary heritage from its sequences.
Does every gene have one of these factors controlling its expression? Are there some genes that are just there, transcribe at will?
Yeah.
I mean, it's hard to, because again, I'm really rusty on my prokaryotes, how they work, but I'm sure that there are genes that are independently regulated. The point of an operon is really about like things that need to be produced together for some sort of biochemical function, right? But there are like housekeeping genes that they must just produce like massive amounts of all the time that probably are not on one of these kinds of like structural motifs. And then again, I think a lot of this comes down to like, how does the cell sense what's in its environment, excuse me, and make changes to its genes to adapt to that environmental condition. So that's fundamentally what all of these like operons and switches are about, is like, how is it sensing its environment and changing in response to that? Does that seem fair, Shoily?
Yeah.
In general, I mean, like both the lac operon and then the earlier one, I'm forgetting which
Tryptophan?
Tryptophan, both were in response to one molecule. Are there operons that respond only in the presence of multiple molecules, or where you got more than one repressor going on, or is it usually this sort of one-to-one relationship?
Yeah, again, my critter knowledge is very rusty. In people, you're gonna see, and you're gonna hear in a little bit, like eukaryotic gene expression is so much messier. So I think the answer probably is yes, but I don't know about it in prokaryotes. I think that's a really good question. Yeah.
What about other, just sort of like, are there like canonical operons that, like the lac operon, it seems like that one's core across prokaryotes. Are there other bags of genes that have an operon like that control particular pathways? Like if somebody asked you like name 5 operons, what would be the 5 you would name?
Probably these.
Yeah, and arabinose probably.
Oh yeah, that's a good one. Yeah, they're mainly metabolism things,
Yeah.
like how does a cell get energy or make things that it needs to be alive. Yeah.
Okay, so I think now's a good time for a break. So maybe 10 minutes. Okay. All right. So let's come back at 6:55.
No.
All right, what's up? So I'm just going to try and kind of pick up where Shoili left off and talk about gene regulation in eukaryotes. Shoot. Okay, so I'm just going to go over my outline, which is I'm just going to talk a little bit about why gene regulation, like what are the unique needs in eukaryotes, some of the experiments we can do to detect or measure gene expression, and then talk about the DNA elements that control gene expression, and then some of the experiments we can do to identify them and to examine their function. So, I mean, the quick and dirty of it is that when we talk about gene expression, we're talking about the synthesis of gene products. We're talking about the synthesis of mRNA and proteins. So that tells us that gene regulation can be controlled at the level of transcription or translation. I am mainly going to be talking about transcription. If you couldn't tell, that's my favorite. And one of the reasons gene expression is so important in eukaryotes is that it confers identity. That's what separates all of the different types of cells that are available in a eukaryotic system. And this is just in an animal system, obviously. You have fat cells, skin cells, what have you. So the amount of a gene can vary in amount, duration, and location, the where and the when. One of the reasons that is important because like we all know that every single cell has the same exact genome, but the unique combinations and amounts of RNA and proteins expressed by each cell are what make it unique and have its unique functions. It knows where to go, it knows what to do, etc. And so I mentioned there's this dimension of space and time, right? So gene expression doesn't vary just between cell types, but it varies between cell states. And it varies a lot during development, which is a lot of what I've studied. And one of the reasons that gene regulation is such an active area of study in eukaryotes. So this is a Drosophila embryo. This is a schematic from like a very classical experiment from like I want to say like the late 80s. Of how genes in the Drosophila embryo change over time and across space. So you have the anterior posterior axis, so one end of the embryo, the other end of the embryo. I'm assuming everyone can see my cursor. Is that true? Yes, okay. And so in all of these colors, we have the different gene names that are expressed in the Drosophila embryo. And this is early in development. This is later in development down at the bottom. So very early, we see these two genes, nanos and bicoid. They're expressed in these like broad regions. We call them domains of expression on either end of the embryo. As we get a little bit later, we see that giant and cruple are expressed again in increasingly specific domains of expression until we get all the way down here to wingless. And some of you might have heard of the mammalian version of that, WNT. In these very specific lines, these very specific domains of expression. So we have our time dimension, we have our time dimension and our space dimension. And when we come to vertebrates, this is a schematic from the vertebrate brain. And I know all of these acronyms are wild looking, but down here where it says TFs, that stands for transcription factors. These are just some of the different genes that can be expressed during the vertebrate brain in the development from a very early neuron, so an immature neuron, we usually refer to those as progenitors, all the way to a more mature neuron. And this is the different layers of the developing brain. And so very early in development, SOX9 and PAX6 are expressed, and we can expect that neuron, that developing neuron, to be here in those bottom two layers. Later on in development, it will express genes like TBR2 and still remain in these bottom 2 layers. Towards the end of its development, it's now expressing genes like TBR1 and it's moved up here, right? So across developmental time, you would not expect, you know, kind of this intermediate progenitor to be expressing TBR1, nor would you expect it to be expressing TBR2, but in the outer part of the developing brain. And so these are some of the messiness that we talked about that happens during vertebrate development. You have your time and space dimensions, and with time and space, gene expression also changes. And then I mentioned cell state, and probably one of the most famous examples of a change in cell state is cell cycle. I assume Shoy Lee talks about cell cycle every single minute of every single day. But during each phase of the cell cycle, you will have a unique set of genes that are expressed for that phase of cell cycle. And so now I'm going to shift a little to some of the experiments that we can do to qualitatively and quantitatively look at gene expression. So I'm going to talk about RNA-seq, qPCR, and in situ hybridization. Immunofluorescence and Western blot are also used for assaying for gene expression, but I guess because the class has already talked about them, there's nothing more I can say. So in situ hybridization is one of the very, classical techniques and is still widely used. In situ meaning in place, and that is because you're looking for RNA expression in the context of the animal or in the context of the tissue itself, the embryo, whatever, the cells. And it was invented in the late 1960s by Dr. Mary Lou Pardue when she was a graduate student at Yale. One of the reasons that this is such a popular technique is that it's very targeted. So we're looking for the RNA transcribed by a particular gene and where we're going to find it. So this is an example that was done by a colleague. This is the chicken retina. So this is the outer part, this is the inner part. And you can see that all 4 of these genes have a unique pattern of expression all at the same time point. So this one is very strong, it's very robust signal all the way up here. Whereas if you look at this guy, THRB, it's in a little bit of that area, but it's also down here in the inner part is very sparse, it's very spread out. The signal is definitely not as robust or concentrated. And that's one of the things that you can see with an in situ hybridization. And how this works is that you have a labeled nucleotide probe that's complementary to your single-stranded RNA transcript of interest. And that nucleotide probe can be made of RNA or DNA. It's usually made through PCR by amplifying the exonic regions of the gene that you're interested in. So if I wanted to make a probe for NeuroD1, I would amplify just the exons of NeuroD1 in order to make my probe. And the label of the probe can be radioactive, it can be fluorescent. There are beautiful pictures of fluorescent in situ hybridization. I think there was actually one in the review article that was shared in the chat on like the regulation. And then one of the very popular ones that's used in this picture is dioxygenin. So the probe has dioxygenin on it, and then kind of by the same principles as immunofluorescence, you can detect that using an antibody.
Can I just ask a question there?
Go for it.
When you're designing the probes, how do you go about knowing which gene to search for?
Well, usually you have a gene of interest in mind. I was scared when you started asking me the question because I've actually never done this technique. But usually you have a gene in mind, right? Like, so these are all, like in this paper, the reason my colleague looked for these genes is because they're known to be expressed in this system at this time point. So they wanted to see what was going on with the expression of these genes. So you have a target in mind.
And so doing it in situ doesn't just tell you that it is expressed, it tells you spatially where it is expressed.
Yes. And as you can see from these pictures also, it tells you how much. There's usually kind of like a baseline control one as well that you can look at to check, did your staining actually work or not? Because in general, working with RNA is a whole can of worms. It's, you have to have like your bench has to be wiped clean with some specific stuff to get rid of the enzymes that degrade RNA. Everything has to be on ice. Everything has to be quick and cold and very, very clean. So you have to have a lot of controls with this technique. But yeah.
So these are actually tissue samples that were then.
Yes, these are developing chicken retinas that were they were sectioned on a cryostat, like so histological samples that were then treated.
Right, then you do the hybridization, the staining, and everything. Yes. Okay, cool.
All right, so wait, any more questions on this? And then just to like keep going on that, like the hybridization part, as I'm sure you've all inferred, refers to the hybridization between the probe and your single-stranded RNA of interest. And this is just kind of a simple workflow of that process. And this is a really cool picture that I wanted to show because this is very similar to that schematic I showed earlier of the Drosophila domains of gene expression. This is all domains of expression of a single gene, the hunchback gene, at different time points in the Drosophila embryo. So you can see how it starts in these very broad domains of expression and it changes at each stage until you get this kind of like dotted pattern, which not a Drosophila person, so I'm not sure, but I think these might actually be individual nuclei that you can see labeled here. So I think it's very cool that you can see with this technique, like both the space and time dimension of gene expression. Okay, so now I'm going to talk about RNA-seq. So where in situ hybridizations are targeted, this is kind of going to be like whole genome. Any possible gene that could be expressed by this cell, by this sample, you're going to see, number one, whether it's expressed or not, and two, you're going to have a quantitative measure of its expression. And so this is, I'm going to talk through this workflow here. Where did my cursor go? So you have your samples of interest. In this example, you have, some tumor tissue and some healthy tissue. You isolate the RNA, and then you're going to generate cDNA, which through a reverse transcriptase, which is the enzyme you buy from a company in a little tiny bottle. You're going to generate cDNA, and then fragment that into pieces, and you're going to add some tags on the end of it. And again, all of this comes in a kit in super tiny bottles that are different colors. You're going to add fragments onto the end of this, and those fragments that you add onto the end, the tags, those have a known sequence. So then you can amplify those, and you can amplify those using primers for, excuse me, the tags sequence, and then sequence those reads as well, because you have the information of the tag. And when you get your sequence back as these little DNA fragments, literally just, AC, TG, blah, blah, blah. And this is sort of the bioinformatic analysis, which I will go through in a bit. You're going to align that to the genome from, like if this was a human genome, you would align it to a human genome, and then start to look for your quantitative data of which genes are expressed, how much, which genes are expressed in condition 1 that are not expressed in condition 2, which genes are more expressed in condition 1 than condition 2, etc. And so I'm actually going to go through an example of an experiment that I've done.
Sorry, could I just interrupt? I missed in the previous slide.
Yes.
The sequence and the black rectangle with all the dots.
Yes. So that's the part where all of this stuff, like this square with like the red and blue and gray, that happens in 250 microliter tubes in a PCR machine. And what you do is you put that on ice and you take it to another university where they sequence it for you.
Oh, okay.
Do is you get on a bus with your box of ice. Try not to look suspicious.
Right. You don't want to give the wrong idea about what you're carrying, right?
Yeah. Yeah, I'm sorry. But yeah, this is something that they have. They have specific machines like Illumina, Model, whatever, NovaSeq, something. They have a new machine every year, and they give you different numbers of reads, literally billions and billions of reads at a time. I think in our lab, we have like asked for like 800 billion reads of like 10 libraries at once.
Wow.
Needless to say, it is super expensive, but every day someone comes out with a new machine that claims to give you more reads for much cheaper. And so I'm just going to talk briefly through an experiment that I've done very recently. So what I study are some of the differences between humans and chimpanzees. So we have our human cells, we have our chimpanzee cells. So, oh shoot, should I be checking the chat? Are there things here? Okay, yes, sorry, I just wanted to make sure I did not miss any questions. Oh, no. So we obtained our RNA reads, like in the whole workflow from the previous slide. And then these steps are just some of the bioinformatics steps we did to align these reads to the genome. And then in R, there's a suite of tools within R. There's a package called DE-seq2, stands for Differential Expression seq2. And that literally, it's exactly what it sounds like. We're looking for the differences in gene expression between our two conditions. And so what that looks like for me is all of the genes on one side of this chart, this is called a volcano plot. These genes are all more highly expressed in the human than in the chimp. All of these guys down here, these pink and yellow guys down here, they're all expressed in more or less equal amounts in the humans and chimps. It's not significantly different in either direction. And then everything on the negative side of the x-axis is more highly expressed in the chimpanzee than in the human. And this is another way of kind of that same information. Every single tiny rectangle represents a gene. This is not an exhaustive list of all of the genes, but every single rectangle here represents a gene, and the color represents the intensity of expression. And that is some of the quantitative data that we have. We were able to graph it in this way. So everything up here is more highly expressed in these 6 samples here, which are actually our human samples. Everything over here is much more highly expressed in our chimpanzee samples. And that's just kind of a little bit about, one possible RNA-seq experiment, what the data might look like, what you can do with it. Questions? Am I good to move to the next slide?
Just a quick question. How do you, I guess there's a prep process, right, where you get the RNA out of the cell?
Yes. So these are cells that we grow on a plate. So we pretty much collect them off the plate, spin them down, freeze them, and then we treat them with Trizol, which smells horrible. We basically burst open the cells with Trizol, and again, in a kit, there's a small kit with columns, and you extract the RNA using that. You have to make sure that you're treating it with DNase. Once again, all of your equipment has to be sprayed down with something that kills the RNA degrading enzymes. And you're basically using a small column to first kind of capture and then to elute off the column your RNA. There is also usually at like sequencing facilities, there's a machine that gives you what's called the RNA integrity number, and that tells you whether or not your RNA sample is degraded and can be used for this experiment. Like it's out of 1 to 10, so I think you want your sample to have a minimum value of 7.
Great, thanks.
I have a question about this slide here.
Go for it.
So in order to do this method with the cDNA and to do this like comparison between chimps and humans, do you have to look at genes that are exactly analogous, or can there be variation between the sequence in the chimpanzee gene and its analog in humans?
You do not have to look for exact homologs. First of all, most of them will be exact homologs because you may have heard the famous statistics that 99% of the coding genome between humans and chimps is identical. In fact, one of the reasons we're doing this is because we're looking for regulatory differences between humans and chimps to understand what makes us different. But one thing we did have to do was filter out genes that have no homolog in one species or the other. Otherwise, when we did this analysis, they would pop up, there would be genes that pop up as being more highly expressed in the chimp, but only because they don't exist in the human. And that would kind of throw us off. So we did have to filter out some genes, but it was based on do they exist in both species or not, and not necessarily are the sequences 100% identical.
And you do that using like informatics approaches, right? You don't do it at some biochemical level. You just like
no, because this is all like when we get the sequence reads to like get the counts and everything, this step where it says obtain transcript counts, that's on the computer. We have the data now back from the facility and we can do whatever we want with it on the computer.
What are some of the key factors that cause these genes to be expressed differently in chimpanzees than in humans and vice versa?
If you know, let me know so I can put you on the paper.
Do you have any guesses, any hypotheses? I mean, what could be different? What could be different?
So I actually didn't put this on the slide because I was like, oh, I don't want to eat up their time with my data. But there's another program you can use which costs $60 an hour to use. And I don't know if that's just my university like trying to rip people off, but it's called Ingenuity Pathway Analysis. You'll find papers say that they did a gene ontology analysis. And there's free programs for this, like Panther and different databases. But the Ingenuity Pathway Analysis is by a company and they have a really great feature, which is they can take all of these things like, okay, these are my human up genes, these are my chimpanzee up genes. You can just put that into the software and it will predict the most likely upstream regulators of this list of genes you've given it. And so we do have some genes that we think are operating upstream of these differentially expressed genes. I don't know if that answers your question.
just for clarification, when you say upstream, you mean genes that regulate other genes that regulate other genes that regulate other genes. You're talking about the ones highest up in the pathway.
Yes, sorry. When I talk about upstream, I'm talking about an hierarchy. I'm not talking about like genomically 5'. I'm so sorry, you're muted.
Sorry. So you don't know what they are yet, but you still think that these upstream factors, the differences are still genomic. They're not like different biochemistry of what a chimpanzee encounters versus what a human encounters.
No, I think this is something inherent about the cells, not about the environment. Like it's not like an ecological environment thing. Although I was reading an essay today that said chimpanzees don't deport people. So that person had some opinions. No, these are about the cells. So this is actually specifically about hippocampal cells. Actually, let me go back several things, because to answer your question more specifically, we think it's this guy, TBR2. Because the stage of development that we're trying to recapitulate with our model is this guy right here, these intermediate progenitors. And we think there are some reasons that we think that humans have some TBR2 binding sites in their regulatory region that chimpanzees don't have. And this may be contributing to differences in the hippocampus.
Cool.
All right. Is there anything else on this slide or regarding RNA-seq? Or you can also ask me at the end. I may not have an answer, but you can ask me. And then the last kind of measurement for gene expression that I wanted to talk about is qPCR. Really, when you're talking about gene expression, what you're doing is qRT-PCR, quantitative reverse transcriptase PCR. To this day, I have a very hard time with the acronyms because qPCR stands for quantitative PCR. qRT-PCR, as I said, stands for reverse transcriptase, but people will also say RT-PCR to mean real-time PCR, which is the same thing as a quantitative PCR, whether or not reverse transcriptase is involved. It's the acronyms regarding this technique are a mess, I will just say that. And so is my understanding that you guys have talked about PCR before, is that correct? Yeah, so this is very similar to just a plain old regular PCR, but instead of any other kind of DNA, you are specifically using DNA, which you've obtained with a reverse transcriptase from your RNA of interest. So like you'll have your sample, you extract the RNA, you make cDNA from your RNA using a reverse transcriptase. And in addition to that, you're going to use a fluorescent dye. And that's what's going to give you your quantitative output from this PCR. And the way you design your primers are you're going to make a set of primers for your target gene. And you're also going to make a set of primers for a housekeeping gene that is, you know, ubiquitously expressed in every single thing. And what the output of that is, it looks usually like this. The y-axis is your output of your DNA, how amplified is your target. So if in your sample you extract the RNA and your target gene of interest is not expressed in that RNA, when you make the cDNA, it's not going to be present. So when you add the primers and try to amplify it, it won't amplify. And what you have down here where it says CT, along the x-axis is the number of cycles. So you'll start to see amplification along with the number of cycles. And one of the reasons you have that housekeeping gene is to make sure that if you don't have amplification, you know that it's not because your RNA extraction didn't work or something like that. The other thing you use that housekeeping gene information for is to normalize your data for when you get the relative expression. What you get out of this is actually relative quantity. So you'll have your control condition, and that is always going to be normalized to 1. And all of your other samples are going to be a relative measure compared to your control condition. Does that make sense? Yes, people are good. Okay.
The control condition is through the housekeeping gene?
No, the control condition is going to be, so for my example here, I have my stem cells versus the ones that have been treated with the differentiation media for 5 days. If you were doing it with the RNA-seq example had like some tumor cells versus some healthy cells. So probably your healthy cells, excuse me, would be your baseline. And then you'd get a relative expression of some genes of interest in your tumor cells.
I see.
But in order to do this calculation, you do need the housekeeping gene. And so now I want to talk about one of my favorite things, control of gene expression. And so this is usually taken over by the non-coding regions. So they don't make proteins, as the schematic says. And these are the intergenic regions. So the huge stretches of DNA between genes, and sometimes also the introns. And overall, these regions are called cis-regulatory DNA. Cis regulators because they're on the same molecule. Yeah, when I was in high school, this was called junk DNA. We had a statistic, 99% of the genome is junk DNA. So we call it cis because it is on the same molecule as the DNA that it's regulating. You would refer to the proteins that bind to DNA as transregulators. And the broad categories of cis-regulatory DNA are promoters and enhancers. And this is kind of how they interact. They go through that looping that Shirley was talking about. Your promoter is where you have RNA polymerase. Your enhancer is where you have all these transcription factors in this big complex. that attracts RNA polymerase and goes ahead and tells the gene to transcribe. So I want to talk a little bit more about some features of enhancers. The general definition of an enhancer or a repressor is that it's just a collection of transcription factor binding sites. And so those bind here, transcription factors, chromatin remodelers, any of the epigenetic enzymes, they will all form a complex here at the enzyme or at the enhancer and recruit the transcription machinery. And your little arrow shows the direction of transcription. And these are all the histones surrounding the area. So keep in mind that this area is relatively histone free. And that's one of the features of enhancers and promoters, is that they are in accessible chromatin regions. They're defined by specific histone modifications. And enhancers can vary in length. They can be as small as 150, 200 base pairs. They can be as large as 2,000 base pairs. And there are squabbles in how much DNA you can consider to be an enhancer. Some people will take a huge 10,000 base pair stretch and say, I found the regulatory region of this gene. They can also be very close or very far. And one really cool feature is that an enhancer, as compared to a promoter, is orientation independent. So promoter is supposed to canonically have the same 5-to-3 orientation as the gene that it's regulating. An enhancer does not necessarily need to. And two of my favorite features are that enhancers are modular. So you can take it out of its genomic context and it's still gonna do what it's gonna do. Like, it has one job and by God, it's gonna do that one job. And they also have spatiotemporal specificity, which is why we make such a fuss about them in development. They will function at a particular time point in a particular location. And these two features together make them really able to withstand mutations without being detrimental to the organism. So because it's so specific and because it can kind of function in and of itself without really affecting anything else, you can have a lot of regulatory mutations and that organism is still going to live. The protein coding sequence is not affected by this. That protein will still be made in all the other places it needs to be made. And that's why they are generally a hotspot for regulatory evolution, for sequence evolution. I see some confused faces.
Yeah, so if the enhancer starts mutating, doesn't that affect what's going to bind to it and then possibly not be as effective in enhancing, in recruiting the transcription machinery?
Yes.
Okay.
And so usually because they're so specific, the organism can actually withstand that. The other thing about enhancers is that there's not, it's not a one-to-one thing. It's not like there's one enhancer for one function. There's a bunch of enhancers per function. Like, honestly, to give you an example, for my thesis, we were looking for an enhancer of a gene, one-cut-one. We screened over 120 possible enhancers, and most of them actually had some expression. But like the ones that we were looking for were like, we boiled it down to 8, like based more and more on like spatial criteria, but around any single gene, you're gonna find, I would even hazard a guess, you'd find thousands of enhancers for any specific gene.
Thousands of enhancers for
Any
just
given
one gene. So you're showing like one enhancer in this diagram, but actually there could be lots of them scattered all around. Lots
scattered all around that are contacting there are diagrams that actually show multiple enhancers contacting a promoter. And so to give an example of the gene that I was working on, one-cut-one, we were looking at it in the retina, so in the eye, but it was originally discovered as being expressed in the liver. So all of these enhancers that we found, some of them maybe they didn't work, they didn't do anything in the eye, maybe they were functioning in the liver. So you could easily knock out one of those, accumulate a mutation, change what's being change the function of that enhancer in terms of what's binding, when it's binding, whatever, you could kill it completely. And maybe one small thing would change in the eye, but the liver would still work totally fine. Have I convinced you all that this is cool?
It's definitely cool.
Can you talk at all about the function of the loop? If the loop has a function that is formed by the enhancer bringing the whole group together with the promoter?
You know, I guess I always thought of the loop as kind of something that needs to happen in order for the enhancer to contact the promoter. Like there are specific proteins that need to be there in order for the loop to form, actually. And there's usually some of these diagrams will show you this big like ring, like a Saturn ring around this entire thing called cohesin that keeps it together. But pretty much the function of the loop is for the enhancer to contact the promoter. As far as I know, I'm sure there are people who study loops and they know all kinds of weird stuff about them. And then the next thing is we want to actually try and identify enhancers or repressors. And the three big ways of doing that are evolutionary conservation, chromatin accessibility, and histone modifications. So evolutionary conservation, the principle there is that if you have a piece of non-coding DNA, so it's not making a protein, but it's conserved in chickens, in marmosets, in whatever, in humans, it's probably doing something. The caveat there is that enhancers are thought to evolve faster than other parts of the genome. So just because something's not conserved doesn't mean it's not an enhancer. And if you focus on conserved regions, you can very easily miss something. So a lot more people these days rely on chromatin accessibility and histone modifications. And so I'm going to talk first about chromatin accessibility. One of the very popular techniques for looking for chromatin accessibility is called ATAC-seq. This is a very recent technique that was first published in 2013. It stands for Assay for Transposase Accessible Chromatin. Transposase just being an enzyme that can kind of freely go and cut the genome wherever it wants, wherever it can. So you have your cells, you burst open your cells in such a way that keeps the entire chromatin intact. So all of your chromatin proteins are still bound to the DNA. You treat that with the transposase. And what that does is it finds all of the very relatively histone-free areas, your accessible chromatin, it cuts them and it leaves behind a tag. And then because we know the sequence of those tags, we can amplify them and make a library the same way we do for RNA-seq. Get on a bus and take it across town to another university and they'll sequence it for you. So these regions are likely to be enhancers because they are available for transcription factors to bind, or of transcriptional machinery if they happen to be a promoter. And when we align that information that we get back to the genome, this is just a little sample of what it looks like. This is from, these two figures are from the 2013 paper where they published ATAC-seq. You see these peaks, and so that means a lot of sequence reads were able to map to this part of the genome, which means this is more histone-free and more accessible than the surrounding areas. And so that's how we interpret the peaks. The big caveat of ATAC-seq is that while it can tell you that these are accessible and there are areas here where transcription factors can bind or chromatin remodelers can combine, what have you, this does not indicate that these are transcriptionally active regions of DNA. That doesn't necessarily mean that these are driving the expression of a gene. It means that they probably can. To look for kind of more direct evidence of transcriptional activity, we would turn to ChIP-seq. ChIP stands for chromatin immunoprecipitation. followed by sequencing. And so this was CHIP itself is an older technique, but ChIP-seq was developed in 2007 and also relies on next-gen sequencing. And it also kind of relies on the same principle of immunofluorescence or like immunocytochemistry in general. We're using an antibody to look for a DNA protein interaction, which is a little more direct evidence. And so that can be a transcription factor binding to the DNA, or that can be a histone modification that we're looking for. So these little red things are points of interest. What you do is you extract the chromatin that you're interested in from your cell population. You're doing it in such a way that the protein DNA interactions remain intact. We sonicate it, which means fragmenting the chromatin using sound waves. Super annoying process. And now you're going to treat your sonicated chromatin with the antibody that you're interested in. And if that's you're looking for transcription factor, your antibody is going to recognize the transcription factor. If you're looking for histone modification, like if you want to look for all of the places where there's H3K27 acetylation, that's what your antibody is going to be for. And then once again, you have a tag on the end, you know the sequence of the tags, so you can amplify the fragments and take them to get sequenced. And this is from a YouTube video. So this person showed how those reads map up against the genome. And questions?
In order to prepare your ChIP-seq, would you usually do the previous, I forget what it's called, the chromatin accessibility piece first, is sort of like narrowing
down what you're looking for? That's an interesting question. That's
what you want to search for?
Yeah, there would definitely be, I would definitely understand if someone took that approach. A lot of times, sometimes people know what they're looking for. People do them in parallel. So people will look for chromatin accessibility, and then they will also look for repressive histone marks and active histone marks and kind of consolidate all of that data together to say, these are my accessible regions. These accessible regions have an active histone mark. These accessible regions have a repressive histone mark. There are also claims that there's a histone mark that differentiates enhancers from promoters. So you can do all of these things in parallel. But if you wanted to, for instance, I think it's on my next slide. If you wanted to, for instance, you can do your bioinformatic analysis first on your attack-seq and say, okay, in all my open chromatin regions, what is the most likely transcription factor? And then do a ChIP against that transcription factor. I think there's a lot of ways you could approach it, and it would just depend on your experiment and what you're looking for. And so this is a little bit about the bioinformatic analysis. And so when you get back your sequence read, do y'all know what a FASTA file is? No, okay.
I may not know it by name, but I'm sure they've seen it. So yeah, definitely walk them through.
Yeah, so FASTA file is, it's a file format that's specific to DNA sequences. I'm gonna put an example in the chat. It starts with a caret and then just, and that's a FASTA file. After that, after the greater than symbol, you can actually put like, so you can be like my sequence 1 and then, and that's the FASTA format. Whatever's in the chat box, that's the FASTA format. The FASTQ format, the Q stands for quality. And so they give some additional information in there to tell you about the quality of the sequence. I read a joke on Twitter that said, I wanted to order pasta, but I wanted to know that it was good. So first I ordered the PastQ. And so that's how you remember that the Q stands for quality. You can then take your FASTQ file, and this is your sequence data and all of your quality assessment scores. And there's a program called FastQC, and that will give you an output like this, where you want all of your sequences to fall within the green zone, or at least the majority of your sequences should fall within the green zone. And once you're satisfied that you have good quality sequences returned, you would then use another program to map them to the genome. And so you have your reference genome, that's like the genome of the organism you're interested in. And each of these is the read, and you just, you ask the program to simply put them together. And that's how you would get like that, the snapshot I showed earlier with the attack seq where you saw the peaks, that's how you would see those peaks. And so when you see words like coverage or depth, that simply refers to how much each area of the genome is covered by the reads. And then you can also take all of this information and use additional software to, instead of you by hand scrolling through the whole genome and saying, I see a peak here, I see a peak here, I see a peak here, you would feed this to a program, and that is the program that would tell you, here is the location of a peak, here's the location of a peak, here's the location of a peak. And based on that, you could identify where your transcription factor is bound or where your histone modification is. If some of them are enriched for motifs, you can do, that's kind of where you can start playing with your data, I guess. I don't know if I've like lost everyone here. No, you're all good? Okay. Questions?
Just this is all about getting to identifying the enhancer of a particular gene. Is that what this whole process would lead to?
It would identify enhancers that exist. If you want to know whether it's for a particular gene, there's different sorts of experiments you would have to do. You'd have to look for enhancer promoter contact. You would have to see that this enhancer drives gene expression in the same pattern that you see in the gene. Yeah, you can look genome wide. That's actually the big advantage here, is that you can look genome wide and look for any enhancer at all. But you can also narrow it down and look, I want to look 100 kilobases this way and 100 kilobases that way of my favorite gene and see what are all the regulatory regions around it.
Yeah, these are typically good ways to get candidates, right? And then you need to follow up with more detailed biochemistry or other like functional assays.
Yes, and speaking of functional assays, one of the most popular ways to look for the function of a potential enhancer is with a reporter assay. And so now we're taking advantage of this property of modularity. So an enhancer is going to drive the same pattern expression within the genome as it would in a reporter plasmid. And so this is an example of a plasmid that a colleague had made. You have your enhancer, you have your promoter, right here you have your reporter gene. And so you can see that these two different enhancers in the retina will drive in two different patterns of expression based on where the GFP is. So, enhancer 1 is only activating gene expression in these cells down here, very sparsely up there, whereas enhancer number 2 here is mostly activating gene expression in these cells over here, some of them down here and some of them down here. And this is my last slide, by the way, so.
I'm trying to understand. So how, when one of the earliest slides you showed of how different genes, or how genes are expressed differently in different cells, whether you're in the eye or you're in a bone cell or a skin cell, how do enhancers contribute to that difference? What is different about cells that makes certain genes express differently in them?
Whether the enhancer is active or not. So let's say you do this attack-seq experiment. Let's say you do the same, you do an attack-seq for, let's go with the cancer example with like this tumor cells versus healthy cells. You would expect to see different enhancers that are active in the tumor versus the healthy cell, or that are accessible. Or if you're say going within one healthy body, if you took some liver cells and some retinal cells, though they both express a lot of the same genes, they both express 1-cut-1 retinoic acid, what they weirdly express a lot of the same genes, you would expect different enhancers to be active in the liver and in the retina.
What factors control which enhancers are active?
Good question. More enhancers, hurdles all the way down.
Ultimately it comes down to what they call like gene regulatory networks, right? Different genes regulate other genes in different places and different times. And it's all ultimately like goes back to like that first stage of development, right? And like the geometry of a body as it's being built.
Yes, some of these enhancers will turn other enhancers off. It's truly a mess, but a really cool one.
I still don't understand how if each cell has the full genome, which is arranged in chromosomes in the same way, whether you're in an eye cell or a skin cell, what is different about a skin cell and an eye cell that causes the genome to be expressed differently? I know that the specific cause is hard, but just generally, what are the different factors?
I would say that it's the enhancers will tell some genes to be off, some genes to be on. And then the genes that are now expressed go and bind to enhancers and tell other genes to be on or off. And then those transcribe more genes that go and tell other genes to be on or off.
So there's an initial signal, right? I mean, you're talking about the process of cell differentiation. There's an initial signal that.
But even the process of cell differentiation is, where's my neurons? My guys, my neurons. This is how you transition between these states is not super well known and not super well studied, and it's not always even the same in every type of cell. So this guy here, PAC6, was originally discovered in Drosophila ileus. In the retina, it opposes the expression of another gene called OTX2. So in cells where you have OTX2, you do not have PAC6. In cells where you have PAC6, you do not have OTX2. And if you knock out one, the other one takes over the organ, right? It's like very clear cut. But now in the hippocampus, PAC6 and OTX2, any cell which expresses PAC6 is almost definitely going to express OTX2. So these regulatory loops, the way that genes control each other, is not even consistent between cell types in the same organism.
Yeah.
Is it right to think about there being sort of cascades that can happen from like enhancers that are really upstream?
Yeah, so there are people, everyone's always looking for the master regulator of whatever. And there are even people who look for, they call them super enhancers, that they're like very, very large regulatory stretches. And the claim is that these super enhancers are serving this master regulator role. That so many transcription factors are binding to this regulatory region that this is setting off the cascade. Caveat being, like my boss doesn't believe in super enhancers. He doesn't think they exist. And then I'm going to tell you another caveat, which I have 10 more minutes to tell you this caveat, that this distinction between enhancer and promoter, some people doesn't think that even exists. There are some people who, one of my professors has written a lot about how enhancers are actually defunct promoters, that the gene that they were supposed to, the gene that they were originally controlling is now controlled by, like, I guess, a better promoter. And so these enhancers are just kind of like secondary to that main promoter now. In the context of how have enhancers come to be. In fact, you can even look for enhancers by, there's a sequencing called RNA polymerase II sequencing. You can look for all of the places where you're getting RNA polymerase II activity and define those as regulatory regions. There's a whole review, I forget who wrote it, but it came out in 2019 just explaining that this enhancer promoter dichotomy is not as clear cut as we've been led to believe, that promoters do in fact have some transcription going in the other direction. Enhancers do in fact transcribe RNA. And they do, it's called enhancer RNA, but no one is sure what it's doing, if anything, or it's just some leftover vestige from when that enhancer used to be a promoter. Cannot stress enough how much the term messy is accurate. I'm also sorry if this answers absolutely none of your questions.
No, it's good. It's good. It's really amazing.
Back to that the sort of super enhancer idea. If that is real, would that be involved in like early cell differentiation? Is that when the super enhancer?
Yeah, I would think that. Yeah, so people will say that they have super, like people have said that in retinal development. So like by the time your retina is forming, the brain has already started forming and sectioned off into like all of its like diencephalon, whatever. So people will say that there are super enhancers that are active at that time point. People will say there are super enhancers that are active in the adult, because enhancers are also relevant for the adult because like, you know, when you respond to stuff inappropriately, that causes disease, like when your cells aren't responding correctly to their environment. Right, they're like enhancers are basically relevant throughout the lifetime of the organism at different states and times. So people will say there's super enhancers for different things. But again, how much people want to believe, like how much evidence there is for that, again, is pretty widely debated because some of these things like the histone modifications don't necessarily support that, for instance. Or people will find histone modification evidence in one cell type, but not the other. It's if you've ever heard of the poem about the blind men and the elephant, it's that.
Are there answers about how a given cell line, say we've been talking about eyes, but when a cell in like the retina divides, does it do something to ensure that its daughter cells will be retinal cells, will be enhancing or will be expressing the same genes it is in the same way?
You should talk to my PhD advisor. You would also like to know. So there's two groups of people, and the answer is always going to be it's a little bit of both. Some people say that it's a stochastic process, that some things will just like by chance turn on one way or the other. And some people say like it's deterministic, like by the time the cell is dividing, it knows what's going to happen to it, right? So if we look, there's some literature in the developing brain that when these guys are dividing, these very early cells are called radial glia, that when they divide, the axis along which they divide is going to determine whether it self-renews to make another radial glia or it moves on to the next stage of development. In the retina, it's all very transcription factor based, like whether it expresses one set of transcription factors, like what's going to happen to the daughter cells. And that's actually that very, I'm sorry, I'm like moving through these slides. I hope no one has where to go. I'm very sorry if you do. So what my colleague was looking for here is there's a type of cell in the retina called a horizontal cell. It's an interneuron. So she was looking to identify, excuse me, enhancers that tell the developing progenitor cell, when you divide, you're going to make a horizontal cell. So the answer is no, it's not no.
Did she find it?
I mean, she found some enhancers. She found some very cool stuff.